In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome to JAX Tech Talks, episode number 69. Today, we are talking through strategies for mouse strain importation and genetic integrity. I'm Adam Waite, Senior Product Manager for the Jackson Laboratory's Cohort Services, and I am joined by Kat Chappelle and Katie Jujin, who are both technical information scientists here at JAX. You may recognize Kat and Katie from previous Tech Talks. Thank you both for your time today. And thank you to our viewers for joining our Tech Talk today. Each talk is a 15 to 20-minute podcast-style webinar where we explore topics that our clients frequently ask us about. For this talk, I've asked Kat and Katie to share what they know about JAX's mouse strain importation, as well as how that relates to clients importing mice into their own facilities and securing genetic integrity long-term. Let's go ahead and get started. The Jackson Laboratory accepts hundreds of strains for donation every year. How often do we find that the strain we get from a donating investigator is not what we expect it to be?
Yeah, so what we've noted is about 25% of the mice that we accept for importation are not quite what we expect. Well, we run into issues with strain background, the mutation itself, additional mutations, genotypes, and sometimes even the breeding for strains that we've accepted. For backgrounds, we're going to see things like an incomplete back cross, undetermined or incorrect substrains. Sometimes we'll even receive a mouse on a totally different background than expected. Mutation issues that we see are often going to be the presence of other constructs, so like neocassettes or Cres. The donating investigator thought were gone during the development of the strain, but we definitely still find them when we test for them. Another big one that we encounter is that the mice we receive are not the correct genotype that the sender believes they are. So we receive a number of strains from DIs who believe their mice are homozygous, and then when we genotype them, we find that the mice are definitely heterozygous.
Oh, wow. Katie, Kat mentioned that we commonly see some instances where something like a neocassette is still there. Can you go into some detail about why that's a concern and how we check for that, and even what our viewers can do on their end to check for it too?
Yes, I can definitely speak to that. Basically, conventional knockout mouse models have one or more genes inactivated in all tissues at all times. So to create such a knockout, targeted allele is replaced by neomycin selection cassette that allows for positive selection of embryonic stem cell clones. If retained, such neocassette can potentially disrupt function of other genes that are nearby. To make sure that neocassette does not cause such off-target effect, most researchers prefer to delete it altogether. To find out if neocassette is present or not in your strain, you can run a genotyping assay specifically targeted to check if a neocassette is still present in your strain.
Sounds good. And Kat, I know you mentioned that sometimes the strain is not on the expected background. Can you talk a bit more about the common issues with background that we find here at JAX?
Yeah, absolutely. So for all the strains that we import into the Jackson Laboratory, we run a custom JAX SNP panel. And that allows us to verify that the SNPs match the expected background. So with C57 Black 6 strains, we often find that there's contribution from both C57 Black 6N and C57 Black 6J, even when the strain is indicated to be congenic on one or the other. We often run into instances where the DI believes that back crossing from one background to another is complete, but our SNP panel will show a loci or two that are still segregating for the original background, and that back cross is not complete. Every once in a while, we get a strain that comes up on a totally different background or contains contributions from different strains that were never really noted in the publication or by the DI. And why this matters is because strain background has important implications in the expressed phenotypes of mutations. So there are a number of strains that exist where the phenotype is more severe on one background than on another. So for a quick example, there's a popular Alzheimer's model that's available on both a congenic and a mixed background. The mixed background exhibits neuronal loss around 6 months of age, but the congenic strain actually doesn't present with this until about a year of age. We also see that on that background, cognitive impairment and synaptic loss occurs a couple months before than the congenic strain does. So that strain background does have important implications as far as phenotype, but it also has really important implications in strain control choice. So while it may not seem like a big deal to use a C57 Black 6J mouse as a control for a strain that has both B6J and B6N contributions, there are documented studies that show this selection does matter and does have an impact on the interpretation of your experimental data. So if you look at the strain tree that we have on this slide, what you'll note is that B6J and B6N kind of form their own branches of substrains. And it's important to realize that these two substrains do carry mutations and other phenotypes that differ from one another. So it does matter as far as control choice goes.
Great to know, Kat. Thank you. Katie, I'm going to switch things up a little bit. Many researchers have maintained their strain at their facility for several generations. Can you touch on the positives and negatives of cryopreservation for us?
Of course. Let's look at the next slide that
Sure.
can help you navigate the advantages of cryopreservation. The long-term maintenance of a strain comes with some risks, like genetic drift or potential loss due to breeding cessation or due to some other worldwide event, like quarantine that we all just have experienced. One of the strategies to address such risk is cryopreservation. The only negative part of cryopreservation that I can think of is that you will have to spend some money upfront to get your strain cryopreserved. But honestly, that amount is insignificant as compared to the risks that you will face when you don't have your mutant strain in cryopreservation. I don't know how many of our listeners are conscious about the fact that every time you breed mice, their genetics is changing through the process called genetic drift. Genetic drift is a real concern as it can cause appearance of various deleterious mutations in the offspring. Such mutations can quickly get fixed in the population, and if not caught early, they can cause the phenotype of your strain to shift, as well as have drastic consequences on the outcomes of your research. In many cases, such negative impact, when it's finally noticed, most breeders by that point in a colony usually are already being impacted. In such instances, if you have cryopreserved your strain, and hopefully you cryopreserved it at some point early in its history, you can recover the animals that will not have such de novo mutations and that will allow you to quickly get back on track with collecting accurate experimental data. The other reason to cryopreserve the strain is that it protects your research against the impact of natural disasters. If your strain got wiped out due to a natural disaster or your facility failure or disease outbreak in your vivarium, then cryopreservation acts as your backup and ensures that you do not lose your strain forever. And more importantly, cryopreservation directly addresses the reduction part of the 3 Rs and improves ethical practices. For example, if you need to put an experiment on hold for a few months, you have a cryopreservation as your backup, you now have 2 additional options that will ensure that the animals are not being wasted due to unnecessary breeding. If you cryopreserve, you can always go back to your cryostock, and therefore you can easily reduce your colony size to just a couple of breeding pairs without running a risk of creating a genetic bottleneck, or you can completely remove the strain from the shelf until you are ready to restart your experiments. Both steps will reduce the number of animals wasted without an immediate impact on your study results. So you will have less issues with labor shortages and free up some space for more other projects that you're going on in your lab.
Great, and really appreciate the graphic. I think this breaks things down very simply. Kat, I'm going to switch back to you. So while the Jackson Lab has more than 13,000 strains available from our repository, we don't have every strain. If a researcher wants to get a unique strain from their collaborator or another institution instead, what are some essential questions they should be asking before getting that strain?
Yeah, that's a really great question, Adam. If you're going to get a strain from a collaborator, then essentially you're going to have to do all of the QC yourself. So that includes getting as much background information on the strain as you can, collect the information down to what vendor they got their original mice from, how many generations they have bred this line, if they've done any instances of backcrossing or colony refreshment, and where the mice for that came from. You're going to want to collect as much information about the development of the strain as you can, such as, is there a potential for unwanted alleles that may still be present, like the Neo or Cree that we talked about earlier? If the strain has bred out an allele at any point, you may want to verify that that allele is actually gone from the mice that you do receive. If you get mice from anyone, even us, you should be genotyping those mice when they arrive. So not only does this let you have confidence in your breeding pairs, it also gives you a chance to optimize that protocol so that it's ready and working throughout that colony maintenance or during your experimental process. If the collaborator you're getting mice from can send positive control DNA, definitely ask for that. That is so invaluable when it comes to having the optimization done and making sure that that protocol works for you. QC for strains is really, really time and resource and effort intensive. So we do this for every single strain that comes into our repositories, just to make sure that, you know, all of researchers know that mutation and background of these strains has been verified. But if you're getting a strain that has not gone through that QC process, you're going to have to be the one to do it. It's a lot of work.
Okay. And thanks again for getting all these questions out the door. There's really a lot here. Wow. Most researchers, though, aren't aware that we offer rederivation services that allow them to ship mice from their collaborator to JAX and have the mice re-derived for importation into their facility. Katie, can you explain why this service is so advantageous for our researchers?
Yeah, this is a great question. And we actually have a couple additional slides that can illustrate pluses and minuses of importing a strain directly to your facility versus sending the animals to JAX for rederivation. Let's look at the first slide. The obvious benefit from having mice to be shipped to you is that you have a full control over what you want to do with these mice. And if you just want a small cohort, it can potentially be at a lower cost to you. The negatives in this case are that these mice have to go through quarantine, which can be as long as 8 weeks or more. In some instances, but by that point, the animals might be too old to be bred. Additionally, many academic facilities have a low health status, so even with quarantine, you can still bring unwanted pathogens that can cause a disease outbreak down the line. The fact that you will need to breed in-house will mean that you or your staff will need to use the time and resources that could otherwise have been used for research. At last, breeding has a lot of variation, and if possible, you always want to get the best breeders at the right breeding age, but sometimes you will get non-productive breeders. And what is worse is that you might not discover it until months have passed by, which can really impact your research timeline. And in some cases, that strain can even be lost. So in contrast, and I say that we switch to another slide, which really helps to illustrate the point that I'm going to try to outline. When you bring your strain to JAX during the importation steps, the gametes from the shipped animals are used for in vitro fertilization procedure, or IVF. The procedures that are used for this step ensure that the animals are recovered into the highest and specific and opportunistic pathogen-free status. Besides, IVF is usually much faster than breeding, and importation to JAX does not require quarantine, which means that fertilization takes place within just a few days of shipment. Additionally, IVF can be scaled to get a very large age-matched cohorts, which are desirable for certain studies. Even in instances when you're starting with heterozygous males, as illustrated in this slide, you can start getting homozygous animals in as little as 2 months. So the bottom line, as seen in this example, if you start with just 2 heterozygous males, you can spend months and months on breeding and still only attain a few experimental mice. While when you send the mice for rederivation at JAX, you can have a cohort of the right size and in best health status in just under 2 months.
Great, Katie, and thanks for sharing another great graphic. I think this breaks things down very easily for our viewers here at home. Well, thank you again, Kat and Katie, for talking us through mouse importation, sharing some strategies with us for keeping those strains secure, not only right when you get them, but also over time. For our viewers, if any of you have questions or comments about what was shared today, please feel free to reach out to us at micetech@jax.org. One of our scientists, maybe even Kat or Katie, will be ready and waiting to help you. Thank you again, everybody, for joining us today, and we hope to see you at another one of our upcoming Tech Talks soon. Enjoy the rest of your day.